Awesome. Thank you so much, Katie. Oh, good morning, Andrew Lyons. Thank you for the good morning to us as well. Unless you're in Eastern time, then good afternoon. Welcome to another installment of the Tennessee Urban Forestry Council's webinar series. As always, I am joined by Kevin Bolger with TUFC, and I am joined by Katie with the University of Tennessee. Katie, I forgot your last name. I'm just realizing it is Donaldson, and it's right there on my screen. So hello, everybody. Happy autumn officially from our last webinar. I'm really excited about our discussion today. We are joined by Bill Finch. For just a quick introduction for Bill so he can get right into all the good stuff. Bill is a founding director, or is the founding director of the Paint Rock Forest Research Center, which partners with the Nature Conservancy and multiple universities to develop cutting-edge forest research on a 4,000-acre nature conservancy preserve in northeastern Alabama. He is the former director of the Mobile Botanical Gardens and was the conservation director for the Alabama chapter of the Nature Conservancy. He is also a founding partner of the Alabama River Diversity Network, a partnership devoted to restoring human and biological diversity along the Alabama River. Finch is an award-winning writer and journalist known for his book Longleaf Forest, The Eye Can See, and his environmental reporting. He hosts a weekly radio show on natural history, the environment, and gardening. And today, Bill will explore how the deep southeastern states and the southernmost Cumberlands serve as ancient warehouses of forest diversity, holding the genetic keys to species we thought were lost through his research at the Paint Rock Forest Research Center. So with that, I'll pass it back over to Katie to do some quick housekeeping for CEUs, and then we're going to get Bill to take it away today.
Right. Good morning, everyone. Please ignore my hoarse voice. I am recovering from a cold. But anyway, if you are on the call today to receive ISA CEUs or SAF CFEs, please make sure that you registered and paid the $10 fee in advance, and that your full name is showing to get credit. And if you need to change it, go to the participants tab, find your name, click the three little dots, and just rename yourself to whatever name you used when you registered. Like I said, if you paid the $10 fee in advance, you don't have to worry about doing that again. Also, if you are a TUFC member, you don't have to pay that fee, or if you're just a general attendee and you're just here for the webinar, you don't have to worry about the fee. Also, if you have any questions during the presentation, please enter those in the chat, and we will get to those after the presentation. And I believe that's it. Imagine, I'll send it back to you.
Groovy. All right. Well, that's all for me. So Bill, if you want to go ahead and share your screen, we can get started with this presentation.
Great. Well, so we thank you all for letting me do this talk. I think I hope it'll be useful to you. We work across the Tennessee line a lot, and we'll be doing more of it. We have agreements with the Nature Conservancy in Tennessee, but we also are doing a lot of seed collection in Tennessee, which I'll talk to you about. I we could have a long discussion about all the problems that are specific to urban forestry. I still deal with those on a weekly basis on my Sunday radio show. And it's something I used to deal with a lot. We are now more into natural forest, wild forest, but I appreciate where you're coming from and some of the challenges you have. Some of the information here will be very useful. I'd like to try to plug that in as I can, but it is of course a long discussion to look at the needs of urban forestry, and and we'll get there as much as we can here. But we're going to talk about our resource and what we have to work from here that we've neglected. And that's one of the things I want you to really think about and look at. So this is the things you need to take away from this talk. I want to talk about species diversity. I got to use all the words that we're not allowed to use anymore. We're going to talk about diversity. We're going to talk about climate and how important understanding climate is, not just the future of climate, but the past of climate. And I want you to understand that. It's hugely important to understanding where you are and why it's so important. We're going to understand species diversity, and but we're also going to understand something else that is incredibly important, and keep it in your head as we go through this. We're going to talk about genetic diversity. And so I want to do a little talk here. Species are a file drawer, and we are moderately good at arranging things in that file drawer, but we're not great at it. And we've missed a lot of things that are very important. And we've missed the fact that we're one of the great centers of genetic diversity in North America, maybe the great center of genetic diversity where trees are concerned. So just keep those things in mind as we go through. Let's get focused on the area. It's that diversity of this area. It's important to look at because that map is, as you can see, a colored mess. And that mess is a very useful to understanding why we're at the center of diversity in North America. It's not the only reason, but it's something to look at. We have huge geological diversity, and that means a lot of diversity in your planning too for urban areas. What may work in some parts of Tennessee may not work in other parts of Tennessee. The diversity is not distributed evenly across Tennessee. That is, there are some areas where diversity, all areas are very high for diversity, but some areas have some types of diversity and some areas don't, and that's because of that geological basis. But there's a couple other things that we need to understand in terms of diversity here. One, we're the center of aquatic diversity, and this is very important for you in urban forestry, even though it doesn't involve trees, because your cities are having a huge impact on this great center of aquatic diversity. This was some work done early on looking at where is the center of aquatic diversity in North America. And it was clear that it was in the southeast, but where was the center of that? And if you look at this map over here in terms of species richness, map A, which is all of the proxies of aquatic diversity combined, it's in a very interesting place. And you'll see it's right there on the Mississippi-Alabama line, a huge red spot. B represents fish diversity in North America. I often say you could go to almost any stream in eastern Tennessee or in Alabama, and in 100 yards of that stream, there's if it's a healthy stream, there are more fish species in 100 yards of that stream than there would be in the entire state of California. Probably more fish species than in the entire West Coast from Mexico up to Canada. The diversity in this part of the world is massive in terms of fish. It is unlike any place else in the world in terms of mussel diversity, right there on the Mississippi-Alabama line. I'm sorry, the Tennessee-Alabama line and into central Tennessee. Um, Alabama, I will have to do this math for Tennessee, but Alabama has more mussel diversity than all of South America. The global center, the global center of mussel diversity is basically mussel shoals, right on the Tennessee-Alabama line. But there's huge diversity there. The center of crawfish diversity globally now appears to be this Middle Tennessee basin. Something clearly is going on here, and things to think about. Turtle diversity. Uh, Alabama is the center of turtle diversity in the Western Hemisphere. I don't think we think about that. We know turtles really well. We run over them all the time. And that's about as much as we think about it. But we're also a great center of turtle diversity, as shown there. We are the center of cave life diversity in North America. And I just want to talk about this. Caves are turning out they're having a big impact on the forest diversity around them that we're just beginning to discover. But those caves in Northeast Alabama and in Southeast Tennessee, that is the North American center of cave diversity. It's not New Mexico. It's not Kentucky. It's right here. Huge diversity of life there, as this study shows. But we're talking about trees. So I want to talk about tree diversity, and some of these things may blow your mind, but it's part of this package of diversity that's right here that we're here to study. And I want you to look at this map. I don't know if you look at the Biota of North America program maps very often. They're not perfect. Some of them are a little out of date, but they give a pretty good impression of where tree diversity is. This is a density gradient of native medium to large tree diversity. Think about where in our heads we talk about, which is Western North Carolina. But then I want you to look at this map. And where is the center of tree diversity? One of the interesting things is that Western Tennessee has more tree diversity than many parts of Western North Carolina, which we think of as the center of tree diversity. But you'll also see, boom, it's much farther south, and we'll talk about that. The density gradient of native broadleaf deciduous trees and shrubs. I don't have to describe to you all what deciduous means, which is a relief. So the center of that, if you'll see, is right there on the Tennessee, Alabama, Georgia line, not farther north where we often think of it. Density gradient of native oak species. This always blows my mind just a little bit. Where did you think that was? If you'll see that it's actually deep in Alabama, and all of this is pretty important for us to understand because of climate and because of climate changes. It's the center of magnolia species diversity. I don't have maps for these yet. We haven't gotten that sophisticated yet, but I want to talk about ash trees a lot because we've got some big problems. But where do you think the center of ash diversity is in North America? I think the Tennessee Alabama line into Georgia comes pretty close to being the center of ash diversity, fraxinus diversity in North America. This is going to be increasingly important. I think we have a lot of maples we're discovering that haven't been described yet, particularly in the hard maple group. So I can't tell you how many maple species we have. We're working on several. But maybe we're the center of maple diversity as well. This is very close to the center of maple diversity here on the Alabama-Tennessee line. Hickory diversity is huge. We have a small area that we're working on the 4,000 acres we're working on. We probably have two-thirds of the hickory diversity globally captured on that site. And there are many sites, particularly in the limestone areas of Tennessee and Alabama, where hickory diversity is just extraordinarily high. And it's not something we think about, but easily Alabama has two-thirds of the hickory diversity globally, and Tennessee probably comes very close to that as well. Hickory diversity is very high, ash diversity. Elm diversity is very high. We're going to talk about that. This was vines, in case you're wondering why you have to encounter so many of those. Those of you who are tree climbers, we're very near the center of vine diversity in North America here on the Tennessee-Alabama line. Trillium diversity. Where do you think that was? It's actually on the Tennessee-Alabama line. And there's a good debate about whether Tennessee or Alabama have more trillium species, but you see where it all converges. Sunflower diversity. Sunflowers aren't native to Ukraine. All the sunflowers in the world come from North America. More than half the species of sunflowers in North America are in Alabama, means that we have about more than half the species of sunflowers globally in Alabama. Tennessee is part of that diversity and comes very close as well. Silphium diversity, azalea diversity is very high in Alabama, Georgia, and Tennessee. Bamboo diversity is actually quite high as well. Alabama is now the center of bamboo diversity in North America. We have our own native bamboo. I hope you're aware of that. I hope you think of using bamboo in urban environments. It's really cool. Not just our natives, but some of the others as well. And there are great ways of doing that. But native bamboo is very much a part of our diversity. And of course, I'll have to say Alabama is the Buckeye state. We have more species of buckeye than any place else, but we're coming up with a new species, which I think Tennessee will share with us. And so Tennessee will come very close to being a center of buckeye diversity. Why is this? Let's look at some of these maps. These maps don't exactly match, excuse me, but just to remind you, there's a lot going on in these areas in terms of diversity of ecosystems and geology and terrain that push that. There's something else that pushes that. Where is the center of rainfall in North America? Mm. It's not New England, and you're getting more rainfall than many places, than Washington, D.C., for example, much more rainfall. We're getting a ton of rainfall in Tennessee, Mississippi, Alabama, that other places just don't get. And we don't think about how important that high rainfall is. It's been very stable for a long time in these areas. And unlike many places like Florida, where rainfall is high now, but it used to be a virtual desert not too many thousands of years ago. There's another thing that we need to understand, which is time. I'm going to try to get this as quickly as I can. That's Ohio. The big white splotch you're seeing is glaciers. And glaciers are a really important element of time that if you don't understand them, you're not understanding your own diversity and you're not understanding our future. Everything in that white line was wiped out by glaciers not very long ago, as time goes. There were no forests in New York. There were no forests in New England. There were no forests in most of Ohio, probably in any part of Ohio for that matter. The Appalachians were tundra and boreal forest, northern Canada type forest, spruce fir forest, almost no deciduous broadleaf diversity at all. And that was, let's give it up to 16, 15,000 years ago. Not when people first arrived on this continent, that's what they were looking at. They were not looking at the forest we see now. So this was fairly recent. Where was this broadleaf forest diversity? Where were all the oaks and hickories and ash and elms and buckeyes? They were farther south, south of that glacial line, south of those impacts. Climate's changing again, and that makes it very important for us to understand, but we also need to understand that climate is not going to be changing in easily predictable ways. That is not how your Christmas is going to look in Nashville. It's going to be a lot more complicated as climate changes. One of the interesting things we're seeing, though, is that climate is changing less dramatically in much of the Southeast than it is in the rest of the country. I don't know how you want to sell that. I don't know how you talk about that, but it's important to understand. And that's a reflection of the long-term stability of climate in these areas. We can look at all of that. The blue areas represent relatively minor changes, or in fact, reverse changes. It's getting colder in spring in some parts of the Southeast. In spring, some of you may have noticed that in some ways. It's some bizarre things happening, but in general, we are much more stable in terms of climate than the rest of the country. And what that means is we're a warehouse. We're a warehouse of species and genetics that don't occur in other places. Let's come back to that native broadleaf deciduous tree and shrub uh, forest and why we decided to build a research center here in Paint Rock because we recognized that we were sitting on top of a warehouse of species and genetics that survived climate change in the past. And we thought that that warehouse was going to be hugely important. We're certain of it now to the future of forest throughout eastern North America because New England, the Appalachians have continually borrowed species from the Southeast as we go through these periods of catastrophic warming, of global warming that have occurred in the past and is now occurring very rapidly. There are 40 species of oaks in Alabama. If you look at the Appalachians from North Carolina, the Appalachian Trail from North Carolina up through New York, there are 12 species of oaks that you'll see if you're very lucky. As climate warms, a lot of those species will be moving north. How they're moving north is going to be a big question because we now have Atlanta and Nashville and Washington, D.C., and a lot of things that weren't there, but they're going to be pretty important. So we decided to come here to Paint Rock, which has been on sort of the leading edge of climate change and climate warehousing of species during climate change for a long period of time. This gives you some sense of that valley. It's very intact. A lot of the forest were intact. The species diversity was incredibly high. We have 150 acres where we have about 100 species of trees, which I'll be talking about in just a minute. The cave diversity was very high, and we now know that those caves are actually playing a really interesting role in preserving that diversity long term. I'm not sure that'll help you in urban forestry, but it's an interesting part of what we're doing here. There are sandstone caps where it's very acid and the soils are very acid. We have a whole community type there that's very distinct. There are limestone slopes that are very steep, where we have all kinds of limestones that accumulated over a couple of 100 million years. They're all very different. We have huge ephemeral flower diversity, maybe one of the great centers of ephemeral flower diversity. We have in virtually endemic species like smoke trees, a really beautiful tree, cotinus, a tree we ought to use more in Tennessee and Alabama, even in urban situations. It does quite well there, and not just the hybrid we're using now. And then we get into this incredible diversity of other tree species. It was all very attractive. So we worked with a guy named Stephen Hubble, who is head of the UCLA Evolutionary Biology Department, Patty Gawadi, the co-chair, and another fellow named E.O. Wilson. Ed Wilson, some of you might have heard of him, probably one of the greatest scientists to emerge from the South, the really the father of modern evolutionary biology. And you know him as through biophilia and other concepts. The very word biodiversity, Ed Wilson popularized. And they all got together saying, we need to have a research facility. What does this research facility look like? We argued about it sometimes brutally for days. And by 2000, we started the program in 2007. By 2016, we were ready to crank the program up. We had funding. We were working with Alabama A&M. We decided this was not just about forest diversity, but it was about training a new generation and making sure that as we train this new generation to understand nature and to be involved in conservation, that we approached underserved communities and made sure they were part of this. So human diversity, a very different kind of thing than biological diversity, but that human cultural diversity became very important to our mission and remains part of our mission. We had 4,000 acres of a very dissected landscape. It's very steep. Our buggy rides are like it's like going to to an amusement park. It's there are a lot of ups and downs. It'll shake loose any kidney stones that you happen to have. But it's a great spot for studying trees and forest diversity because of all the aspects, because of all the slopes, because of all the different exposures of geology. And on that 4,000 acres we have with Nature Conservancy, we'll be adding to it shortly. You'll see a rectangle down towards the bottom that's embedded in that. That is our 150-acre, 60-hectare forest plot where we have 90,000 trees tagged, mapped, measured, identified, and they'll be followed for 50 years. It's part of the Forest Geo Network. It's the largest outdoor laboratory for trees in North America. It's really astonishing, and it's astonishing how many people we have working there now. I'm surprised. I'm trying to coordinate them all. It gets to be difficult. We have more proposals. We're studying everything from forest history to carbon to hydrology. We're looking at new species all the time, and we have a campus, a very comfortable campus for all of our researchers, where all of our researchers can stay. The process of developing the 60-hectare plot took many years and many people over time to develop it. Uh, and uh, and there you have it. Each of those 90,000 stems has a tag, its own individual number. It is an effort that you can't imagine until you actually see it. If you can imagine packed 100 about 125 football fields packed with trees, every one of them identified, every one of them with a tag. It's really quite remarkable. And we've been able to attract researchers from all over. And this is the result of some of that work. The map on the right represents every stem in that 150 acres. We know where each one is. We know what geological exposures it's on. We know why it's there. On the left, that very colorful graphic is another way of looking at distributions. One of the things we're finding is that species aren't evenly distributed across the landscape. They cling to certain types of areas, and this is will become very important for you in urban forestry, but also very important in understanding what kind of forests we need to preserve in North America. Our cave research has really developed with Hazel Barton, who is now looking at, she's at University of Alabama, one of the world's top cave researchers. She is now looking at photosynthesis in the dark parts of caves. That's a whole nother subject. I will let you dwell on that for a minute and figure that one out. But we're now beginning to understand that caves have an important role in carbon cycling and in nutrient cycling in the forest that surround them. And we have taxonomists from all over who are exploring diversity within that forest. We have a new species of azalea. We're working on a new species of buckeye. We have a new species of violet, which I'm not supposed to be showing you till we publish it. We are going to be looking at if anyone can identify that oak. It's in Tennessee. We're seeing it more and more in Tennessee. It's a new species of oak. We've done the chemistry on it. It's really interesting. It's really quite widespread. It's hard to understand why people have overlooked it. There's many weird excuses. Sometimes claiming that oaks hybridize is sort of the refuge of cowards who don't want to look at the taxonomy. But yes, oaks do hybridize, but species sometimes result from that. And as long as they're reproductively isolated and maintaining themselves as species, they're good. So we got a new oak species. And then I want to talk about the elms real quickly. You don't think about it, but Tennessee and Alabama is the great center of elm diversity. I'm pretty well convinced in North America. And it's not just the trees, that is American elm. And if you grew up in much of Tennessee and if you grew up in Alabama and you kept hearing about Dutch elm disease and you said, gosh, I wonder when the Dutch elm disease is finally going to take out all of our elms. Because most of the places I go in Alabama, they're elms, American elms, and they're perfectly healthy. And they don't have Dutch elm disease. In fact, they're one of the dominant elements in many forests. And we have elms that people don't talk about, like September elm. And by golly, you folks in Tennessee should be talking about September elm like we should be talking about September elms. It was part of a lot of our urban landscapes for a while. People actually talked about planting September elm in Chattanooga and in Birmingham and in Huntsville. And they were proud of those. You don't know what September elm is? Go look it up. It's quite distinctive and a part of our ecosystem. But let's get back to American elms because why are our American elms just surviving here when we lost them by the millions in New England and in North Carolina and in New York and in Michigan? What happened? Pennsylvania. What happened? Well, let's look at climate and let's look at that ancient warehouse of species. What species managed to sneak north? Why did the species that got north after the climate changes, why did they get there? Maybe they were better adapted to cold. Maybe they had a lot of features that helped them move in behind the glaciers. Maybe they were relatively naive to diseases because there weren't a lot of forest diseases in areas that were being pioneered after the glaciers left. And so the trees, the American elms that went up north, were actually polyploids. Better not get into that, but they were polyploids. They had special features that allowed them to survive that. But the elms in the south, diploids and polyploids, uh, weren't necessarily limited to those features. And so we had a huge number of elms that survived here. So if you're reading information about why you shouldn't be planting elms in your cities, that's coming out of New England. You need to be looking at your own forest and the genetic diversity that's here. We now know that within American elms, we're probably going to end up with two species and maybe three species. Two of those species we know have been separated by about 11 million years. That's how closely we're looking at our forest, is that we really don't know the diversity within our species groups. Elms are really great. Phytophthora is another big issue. Oh, am I going to run out of time? We're seeing this more and more in our landscapes. A lot of trees down south have managed to survive Phytophthora. Some didn't. Franklinia is one. Chestnuts, as we're discovering, it wasn't chestnut blight that wiped out chestnuts throughout much of Tennessee and Alabama. It was, in fact, Phytophthora, a root rot disease that we've forgotten about. And one of the things that I think we're going to see in these southern forests is that they've been through the keyhole of Phytophthora, and they're better resistant to that. And Phytophthora now is moving northward and impacting forest much farther north. That's why we're working with Hudson Alpha to look at the genetic differences between trees. And as, for example, the emerald ash borer comes down, we have a huge diversity of ash trees. We have three species of what we used to call white ash in our forest. We also have blue ash, we have green ash. How are these southern trees, not only because of the diversity of species, but because of the diversity of genetics, are they going to be able to better survive the emerald ash borer? It remains to be seen. But we want to know some trees will survive. We want to know the genetics of the trees that survive. And we want to know, just as importantly, the genetics of trees that didn't survive, because that's the only way we'll know which genes are important. So we're doing huge genetic work with ash, with walnut, which is beginning to suffer from diseases, beech, elms, uh, and many other trees to help understand what are the genetics that are going to survive into the future. Um, and I want to talk a little bit about climate change, just looking at some of the maps. This is the current range of live oaks, just to give you a sense as climate begins to warm, which it will inevitably do. And it may happen relatively rapidly now. I'll let you decide how rapidly that happens. But that is the potential range under a very foreseeable future under Hadley 8.5 of live oaks. Kind of amazing, isn't it? Another tree that we need to focus more on is shortleaf, and it's something we're focusing on a lot. Shortleaf is the longleaf pine of Tennessee. It was hugely important to large swaths of Tennessee in terms of the kind of savanna habitat that it created. I'd love to talk more about savannas, but that is a savanna there. It's a forest with grasslands underneath it. It's something we're studying more and more. We've forgotten about shortleaf. Bowater brought a lot of loblolly to Tennessee, and it's beginning to overwhelm that shortleaf resource. But shortleaf was the better tree in many ways and was part of an important ecosystem. We're now trying to restore that, particularly as loblolly is struggling. But here's looking at how resilient shortleaf is. Shortleaf had a huge range and extending well up into Long Island, but and back towards Missouri. But that range is likely to increase as climate warms. So we think it's a great tree of the future and one we're doing more work on. Unfortunately, no one has collected seeds from eastern shortleaf because we have so little respect for our own forest. No one's collected seed from those trees in almost a century. That's something we're doing now. I hope to talk with you all more about that because we definitely want to collect from more trees in Tennessee. I will tell you that collection requires that we collect green cones because once those cones open, they have shed their seeds far and wide. That's why they have those little wings. And there's the problem. How do you collect cones from the tops of trees? I will let you all imagine how that works, but these are on the very tips of branches, which means it's very difficult to climb. I used to climb, I'm too old for it. But it's how are we going to do that? We've come up with many tools for doing that. We're using arborists a lot with bucket trucks. We use Hastings poles in many cases where we can reach, which requires its own effort. But we have collected about 2 million seeds. We're putting those into production. We're going to have a great shortleaf year here. And this is our own production equipment where we actually produce those seeds and we send them off for production as seedlings. We're actually creating orchards too, where we can do that. So it's a big part of our mission as well, and love to talk with you more about that shortleaf component. So this is kind of a brief overview, as brief as I could make it, of the work we're doing at the Paint Rock Forest Research Center. And I want to challenge you to think very carefully about the diversity in your own backyard. We have wiped it out in many cases. We don't have a lot of respect for it. We tend to imagine it occurring in places different than where it's actually occurring. We have very little protection for areas where the diversity is highest. And many of the areas that are protected are the areas where diversity is relatively less. So we've got a lot of work to do. I also challenge you to think about when you're using trees in your urban forest, please think about using local genetics. It's hugely important, not only for the survival of those trees, but for maintaining this warehouse, this hugely important warehouse that will be important to all of Eastern North America. I think I can end it there and open it up to questions. Let me get out of this. There we go.
Well, thank you so much for that discussion, Bill. That was very interesting. I would say, at least on my part, I don't feel like we have people like you speak often enough to urban foresters and a lot of us like arborists. And so that was a very interesting discussion. We do have a few questions, and I also have a few questions myself, but I'll let everybody else go first. Katie, did you want to handle questions at this time?
Yes, and one thing I forgot to plug in, we have a national shortleaf conference coming up November 11th in Gunnersville State Park. It's a 3-day conference. We're going to be touring some of the areas where we're looking at shortleaf and trying to restore them. We're going to be talking about shortleaf as a major tree, something we've got about 200 or 300 people from all across the country coming there from Missouri to the Carolinas and a lot of folks in Tennessee. I encourage you all to come and see what we're doing with shortleaf. There will be some opportunities to plug a lot of you arborists into what we're doing in Tennessee, and I would love to do that. So we'll be talking more about that as the time comes and as we near a major seed crop, which I think we'll do in the next 2 years. So I'd love to have you all involved in that. Be sure to look that up, Shortleaf Pine Initiative. Shortleaf Pine Initiative. Go look that up on the internet and you'll find the link to the conference. Shortleaf Pine Initiative. I'm ready.
Okay. So first question, this is from Sarah Wells. She said, if I understood correctly, what is the evidence that chestnuts succumb to Phytophthora instead of blight in the Southeast?
Yo, it's really cool. I'd love to talk about that more. The evidence is that a lot of this, one of the latest pieces of evidence is that a lot of the chestnut that people planted out died of Phytophthora. And so now we're having to go back and rethink chestnut genetics because it wasn't just the blight, it was Phytophthora. We had early accounts from oh gosh, what is his name? Harper, Francis Harper, and Roland Harper both had accounts in the Southeast, and they were looking at the southeastern forest as they were beginning to die, as the remnants of that chestnut forest were beginning to die from chestnut blight shortly after the turn of the 20th century. But they said, look, all the old timers said that we lost chestnuts, most of our chestnuts, in the 1870s, and Harper was a bit of a genius. Roland Harper said, I think it was probably what we call watermelon root rot in the old days, which was cotton root rot. It was a Phytophthora that had been introduced, Phytophthora cinnamomum. And as it turns out, now research is showing that is indeed what happened. And so a whole suite of species were impacted, including Franklinia, by the introduction of this Asian Phytophthora, which didn't survive the cold very well. So in the Appalachians, where the ground would freeze, where it used to freeze in winter, Phytophthora didn't exist there. So the trees survived. And people had the impression, oh, well, because we started recording it in the late 19th century. People said, oh, well, this is the center of chestnuts. No, it wasn't. It was much farther south, but they had been wiped out by Phytophthora. So there's plenty of evidence now. And the people who are trying to redevelop a blight-resistant chestnut are now having to redevelop chestnuts that are resistant to Phytophthora as well as some other diseases. So it wasn't as simple as chestnut blight. Is that a sufficient answer?
Yes, thank you.
A lot
Next
of
question.
explanations for that, but we won't have time to talk about them all. Yeah.
Next question. How do you find trees that are genetically local? Do you garden nurseries, tag them as such? Or
Yeah, so that's really important. I think we need to explore our own forest better. One of the problems I have with shortleaf, and this is a really big problem, is that when people decided shortleaf pine was an important tree, they were, as usual, we were pretty lazy about it in the Southeast, excuse me. So we got all of our seeds from Arkansas. Turns out those Arkansas shortleaf are actually hybrids, in many cases, of shortleaf and loblolly. It also turns out that the shortleaf from Arkansas tended to be more like loblolly than eastern shortleaf. And so we began overwhelming the genetics in Tennessee and Alabama with our restoration efforts with these seeds from Arkansas. And there's nothing wrong with Arkansas. It's a great place. I highly recommend it. But you don't want to get your seeds from Arkansas. You want to be getting your shortleaf seeds from Tennessee. Uh, and and that's one of our efforts is to make that collection. How do we figure out whether we have native shortleaf or not? It's a big question. We can go into a lot of discussions. But when we're restoring, it drives me crazy when we take uh, cherry bark oak from the Louisiana bottomlands and try to plant it in Tennessee. And that's what we often do. We have our own cherry bark oaks in Tennessee. We ought to be using those appropriately in Tennessee. We don't want to get our American elms from Ohio, from stock up north, which is what we've been doing, which is why they're failing. We want to get our American elms from stock in Tennessee, which is one of the great centers of Dutch elm disease resistance in the Cumberlands. So looking at these native forests, finding sources that are good, collecting the acorns, collecting the seeds is hugely important to what we're trying to do ultimately.
And if I might ask on that note, do you and does your organization do anything like currently to distribute seeds to nurseries? Or are there any initiatives that like you're doing in partnership, like in the state of Tennessee specifically, or in Alabama to support that?
Yes, so I did talk to Tennessee State Nurseries about the shortleaf, and uh, I won't go into that. We've been in discussions for a couple of years, and I think something will come of that. And I'm very hopeful. We are not yet in a position to distribute uh, broadleaf seeds. We really haven't developed that enough. We don't have enough people, time, and money to do that. But it's something we hope to do in the future. And it's something I encourage you all to do. And there are many ways of doing that. I mean, there's nothing wrong with collecting acorns from native forest and growing them out at your botanical gardens. Kind of a great project for people. Acorns are very simple. Hickories are very simple. They might need some vernalization. They might need some, chilling hours, but it's very easy to do. And this is the kind of thing we should be doing. You need to encourage your cities to I don't want to get too deep into urban forestry, but I could. You need to encourage your cities not to use these giant trees when they're planting, because inevitably those giant trees, not only do they have poor success and poor rooting, they're going to have lots of failures, but inevitably they're going to be genetically depauperate. They're going to come from God knows where. We're creating a disaster by planting these large trees. We need to encourage cities to think about ways to planting smaller trees, which are going to be much easier to grow out from native sources. You don't have to have special resources to get a 3 or 4 inch caliper tree. So all of these things are really important as we begin thinking about restoring our native forest, is how many we've trapped ourselves into using a very small number of species and a very narrow genetic range within those species because of some of our requirements in urban environments. Sorry, I hope I didn't step on any toes.
To go with that, this next question, is there a list of species that you have curated for urban areas to create the genetic diversity that you're emphasizing in Tennessee or Alabama?
I've done this list for South Alabama. I worked on a list with Thrive in Chattanooga. It wasn't as big a list as I would like. I would love to develop that for Tennessee and begin thinking about it. Urban environments have a lot of special requirements because they're often low oxygen. The conditions are actually much like the trees that do in swamps actually do better in many urban environments because of the kinds of conditions we create there, the compacted soils and other things. So it's a very specialized list for these urban environments in many cases. But I would, it'd be fun. If you all decide to develop a list someday and let's brainstorm that, I'd love to work with you based on what we're seeing. I can sort of give you the range of species and the types of soils that they're working with. You can begin thinking about your environments. I'd love to do that sometime. I don't know of a list in Tennessee that really does that.
Um, this next question, what are some of the questions you are asking of your 90,000 tree database?
Yes. So the first thing we have is Hudson Alpha coming, and we're actually this is so cool and it's so bizarre. I can't believe we're doing it. It's going to be so expensive. But we're looking at let's take every fraxinus, let's take every ash tree on the plot, and let's look at the DNA in that plot and that in those trees, every stem. And that's what we're doing. It's insane. we're going to look at persimmons, which is going to be really fun because we see there's a huge diversity within the persimmon group. It's not immediately threatened by disease. I'm sure we're going to find one to import soon and threaten persimmons with, but right now it's okay. But the diversity within persimmons is really cool. And we're sitting on this, what I think is a real gold mine. It's the best tasting persimmon in the world, as far as I'm concerned. It's just that we haven't developed it. How do we develop it? What is the potential there for thinking about it? That's one way of thinking about it. But ecologically, we're doing some very important things because one of the things we're finding, as I mentioned, is that trees aren't evenly distributed. We find that maples, this is bizarre, maples and beech don't co-occur. I mean, rarely, but almost never in our forest situation, and we can document that now. Why in the heck is that? Why do hard maples and beech rarely co-occur in the Cumberlands? I don't know. But you know what? If you're trying to do forest conservation, and you better know it. If you're trying to decide what kind of soils beech and maples should be growing in an urban environment, you better understand that. So understanding why trees seem to be affixed to certain areas is really important. It's not just hydrology, it's not just water availability. We've pretty well been able to eliminate that in many cases. There's some other factors. And I think one of the big things, and we're just on the cusp of studying that, is soil communities. And there's 10,000 species we don't know in the soils on our preserve. We don't know them. We don't have names for them. We don't know what they are. We don't know how they interact. We don't know how they're affecting trees. But just like Phytophthora played a huge role in chestnut demise, it's a soil disease. We suspect there are a lot of other things happening, oomycetes, bacteria, fungi that are affecting this forest community for good or for ill. We tend to think really nice things about fungi. Oh, isn't it nice that we have this cozy relationship with mycorrhiza and all that kind of stuff? That's all fine. But there's some pathogenic stuff in there too, and we don't know how those things interact. So our next step is going to be to try to determine what is that interaction between soil communities and trees. And we'll kind of have the perfect place to do it because we have so many soil types and because we have so many trees cataloged, and it's just going to be enormously expensive, enormously difficult, but really fun.
Okay. Next question. What are your insights on crepe myrtle, invasive, native, aggressive?
Oh gosh. Let me be nice. You know what? Your crepe myrtles, I don't have to pronounce anything on crepe myrtles. Your crepe myrtles are about to be hit by all the things that are hitting crepe myrtles in the South because why? We can only think of one tree at a time. We're sitting in the center of forest diversity. We can only think of one tree at a time. We only plant one kind of tree with very narrow diversity, and it's even been selected down so that it has even narrow diversity. The pathogens that are affecting crepe myrtles in the south, which are making in the deep south, which are turning them into ugly black sticks, are coming for you. So be prepared. Uh, your crepe myrtle forests are doomed. We're having problems with crepe myrtles being invasive in certain sites in South Alabama and in Central Alabama. That may become a problem farther north as well. But there's a lot of diseases coming too, and I think it'll take care of that as well. Just be prepared.
Um, what is your experience with planting longleaf pine in the northern part of the historical range of this tree?
Within the northern range, up into Cherokee County, Alabama, and Rome, Georgia, it's fine. I think you're wasting your time to plant longleaf in Tennessee right now when we have this incredible shortleaf resource, which is much better adapted to conditions that doesn't look. I wrote a book on longleaf. I love longleaf, but it's not where Tennessee should be. If you want to have a kind of a interesting ornamental, that's fine. But from a forest perspective, we should be looking at shortleaf. It's just clear. If you have an ice storm, you got big problems with longleaf. You're going to have much fewer problems with shortleaf, and that's just one of many examples. So it's the shortleaf community that I would encourage you to focus on. Please believe me, I wrote a book on longleaf. I love longleaf. I grew up with it, but you should be thinking about shortleaf.
Bear with me. I'm just trying to keep track of all these questions. Um, as you evaluate all these multiple variations of the same species, will there be an identification guide?
We use the flora of the southern United of the flora of the FSUS, flora of the southern United States, which is Weekly's book out of North Carolina. I work with Alan. Alan, we were supposed to have a call this week to discuss some of these new species. Generally, as we come across new species, they are recorded as what we think are new species, even if we don't have all the genetic work done. I discussed that with Alan, and he's beginning to record them. So Quercus species one, if you want to know what that looks like in eastern and central Tennessee, east of the Nashville Basin, you can go to species one in Alan Weekly's Flora, and you can look at the key there to see it. We don't have the maples sorted out very well yet. We got a lot of work to do there. The elms, so I think that's the resource right now, and that's where I would put it because it's refereed, and you don't have to take just my word for it. So the flora of the southern United States, when we started this program, one of the universities brought Peterson field guides from the 1960s, and I made them throw them all away. And this is one of our problems here is that we're using field guides from the Northeast that were written decades ago that have no comprehension of diversity here. And even field guides from the eastern slopes of the Appalachians have no concept of the diversity on the western slopes of the Appalachians. So be very careful and cautious about that. We need to be doing more work, but right now, the flora of the southern United States is our guide, and it's pretty good.
I was going to say, if you're looking to author a second book, I think most people here would probably purchase yours if you decided to come up with one for an ID guide
Yeah, gosh,
with all the
I'm
time
getting
I'm sure you
old,
have on
folks.
your hands. So,
I'm getting old. So, yeah, it won't be me, but maybe we'll train a new generation to do that.
and then one other thing before Katie moves on, just to interject for those of us that are looking to kind of read into some of the published research that you and your organization have done, what sites or journals or places can we go to kind of learn more about the research that you're doing? Like, where are those places?
Yeah, so we started this in 2017. It took us a long time to get the census done. We are just now beginning to publish the first phase of our census data. So, and it will be in multiple publications. One's in forest. We got some new stuff coming out in forest ecology. We've got some stuff coming out in Bretonia, or which is now not Bretonia, but something else, uh, which is the new violet. You know, it's going to be in a lot of. Some of it's going to be in Castania. We found an extinct tree, one of only the only five trees were declared extinct in North America. The only one that was truly extinct in the wild we found. And so we're going to publish that. It's a really interesting story. And we'll publish that in Castania coming up this coming year. The process of publishing things in academic journals is painfully slow. We're getting there.
I do have just a couple more questions left. Everyone, I know it's 1 o'clock, so if you have to get off, feel free to jump off the webinar. Kevin did post a link where we can record your attendance. That's just an extra step to help us if you're seeking those CEUs. Um, but would you mind just answering a couple of more questions?
I'm here.
Okay, great. Um, will your research be able to demonstrate species-specific mycorrhizal data into mapping network format?
Yeah, wouldn't that be great? It would be great. I need a few $1 million, but yes, if you can resurrect NSF for us, it'll help. We're in a, it's a rough time, and we need to acknowledge that. A lot of researchers are really struggling now, and a lot of researchers I would be working with to do that are having a really hard time finding funding right now. It's a really serious, serious time. Uh, I, yes, we would love to do that. That is a major work. It's even with the money, it can take many years to develop something like that, but it's something we'll be certainly working towards.
Um, This question, can groups come down to your location for a talk and tour, and how would we arrange that?
Yes, so let's do that sometime. It is a rugged landscape, so to get around, we need buggies. Uh, it's in a big landscape, but we could certainly, for your group, I think you're an important group for what we're doing and for disseminating this knowledge and for using this knowledge. We would certainly do everything we could to accommodate you for a tour. Just recognize we need to do a little bit of planning and figuring that out. I'd love to have you all here.
I'm going to thumb mark that for the 2026 plans, and we will definitely be continuing that conversation.
All right, well, I don't want to keep us too far over, so imagine I'm going to go ahead and have you wrap it up.
Yeah, absolutely. So, again, thank you so much, Bill. This has been riveting. This has been such an interesting discussion. I can speak for everybody, I'm sure, on this webinar. I don't think this is the last time we're going to have you out because this has been really great. So I'm so grateful you were connected to us. For those of you that aren't able to potentially go to the conference in Alabama in November, just keep in mind TUFC also has the annual conference the 12th through the 14th in the city of Chattanooga for us as well. So if you can't make it out to Alabama and you can make it to our TUFC annual conference, we'd love to see you out there as well. And I think in the future, Bill, we're going to have you back out. So I look forward to it. Thank you all so much for your time. Thank you so much, Bill. Thanks, Kevin. Thanks, Katie. And have a great day, everybody.
Thank you.